Well, thanks, Josh, for that really kind introduction. I'm really delighted to be here. My family roots are in the Midwest. It's always great to come back to the heartland. I've never been given a talk at WashU before, and so I'm really pleased. And I'm pleased that we have this kind of concentration today in something that's very close to my heart and professional interests, which are the comparison of males and females in the study of sex differences. I'm also really pleased that you put me on the lineup today before Peg McCarthy, because she gives such a terrific talk, and I'm glad to kind of slip in before she raises the bar. I think we're going to kind of have a little bit of a divide here. I'm going to talk a little bit more about sex chromosomes, and Peg's going to talk a lot about hormones. And the hormones are usually more important than the sex chromosomes, but it all starts with the chromosomes. So, okay, I want to give a plug for our journal, The Biology of Sex Differences, which was founded about 5 years ago. It's the official journal of the Organization for the Study of Sex Differences. The OSSD has annual meetings every year, which are really terrific. And if you're interested in sex differences at all, that's you hear all the latest and greatest there every year. Next year, it's going to be in Philadelphia. If you submit to our journal, it's an online journal, and the articles are available for free to anybody who has access to the internet, so we invite your submissions. There are 4 points I want to make today. The first is kind of the politics of studying sex differences and changes that have occurred recently in that area. Comparing the sexes is really the right thing to do, and it uncovers some really terrifically interesting science. Second of all, I want to try to establish this idea about thinking about the XX and XY cell as having evolved to deal with these different sex chromosomes, which bias the cells. And partly evolution has tried to make XX and XY cells more similar. But that's hard to do because they start out different. Thirdly, I want to talk about some evidence that the number of X chromosomes, in particular, makes a difference and causes sex differences in adult phenotypes related to disease. And the fourth point is that a point that will run throughout the talk is that we have these compensatory or offsetting actions. That you have sex-specific processes that counteract each other and actually make the sexes more similar. But that if one of the processes gets disturbed by disease or by changes in hormone levels, then the other process may cause a sex difference to emerge. All right, so first of all, number one, so we start out with the observation that many, perhaps most, perhaps even all, but most diseases have a sex bias. That is, they occur more in one sex, or they're worse in one sex, the progression is more in one sex. These are some neurological and behavior disorders. I'm going to talk a little bit about multiple sclerosis, which is down the list. It's about women get MS 3 or 4 times more often than men. But when men get it, it can be worse in some cases than in the women. So it's a complex sex difference. The incidence is certainly female biased. And so we'd like to, just as a fundamental way of understanding this disease, we have to deal with the sex difference. If you don't understand the sex difference, then you don't really understand the disease. But biomedical science is sex biased. I'm going to talk about a couple of heroes. One of my heroes is Irv Zucker, who's a retired psychologist from UC Berkeley, and he and his postdoc published the study in 2011 where they just counted papers in 1 year, 2009, and just went through the literature in a few journals in each field and said, okay, how many, these are animal studies, how many papers studied just males, how many studied females, how many studied both, and how many you couldn't tell because it didn't tell you. So the gray bars, look at immunology. 3/4 of the papers in immunology don't happen to mention, okay, whether they studied males or females. Like, how stupid is that? But look at neuroscience. Let's see, do I have a pointer? Is this? Oops, this is, okay, sorry. Oops, no, this is the forward, and then this is the. And then this is, okay. So if you look at neuroscience, the blue bars are the male-only studies, the yellow bars, part of the bar is the female-only studies. It's like a 5 times more neuroscience papers on males than females. So we have a significant problem here, and that is that the biomedical literature may apply more reliably to males than females. Now, why are males studied much more frequently? A dominant reason that people, a lot of neuroscientists that I know don't want to study females because they're afraid of the estrous cycle. They're afraid of this variability, and that these unfortunate hormones that get in the way, okay? And they just don't want to deal with it. I'm interested in synaptic physiology. I don't want to have to worry about whether the female's in her estrous state or diestrous or what is she, what's her hormones. I don't want to deal with that. I just want to understand the fundamental thing about synaptic physiology. Well, okay, so our hero here, Irv Zucker, and his former student Brian Prendergast, who's a professor at University of Chicago, did a study where they went through a whole bunch of papers. They went through 293 articles, okay, measured almost 10,000 traits, and they measured the variability of these articles all, in fact, compared males and females on traits, and they measured the coefficient of variation. So you have to measure the mean and the standard deviation of every trait, which they only could do most of the time by measuring the histogram bars and the error bars, and then calculating the coefficient of variation. And to ask, okay, well, and these are gonad intact males and female mice, okay? If the females are more variable because of their unfortunate hormones, then you should see, this is a histogram where they have when the female variance is greater than the male variance, or the coefficient of variation, then the study or the trait appears as a bar on this side of the curve. And if it's the opposite, the males are more variable than the females, it occurs on this side of the curve. And you can see it's about even Steven, okay? There is no sex bias in variability in the trait, okay, that they could pull out looking at hundreds or thousands of traits. So kind of not to be picking up on this idea and kind of in a copycat study, which we're just now publishing in the Biology of Sex Differences, my colleague Yuichiro Ito went through, so you say, where can you get a lot of traits that you can measure? Well, you can go to gene expression databases, microarray databases, which are in Geo, online, and you can just pull out, in fact, Yuichiro pulled out 293 studies measuring over, with 16,000 probes per study or something. And so he looked at the variability of 5 million probes over many, many studies to say, okay, is the variability of gene expression greater in females than in males? And in fact, the answer is no. So in humans or in mouse, again, so here, if the variance is higher in males, it occurs on this side of the curve. If the variance is higher in females, it occurs on this side of the curve. And it's a pretty even distribution. Actually, it's shifted towards greater variability in males, and because the N is so high here, you can get a statistically significant shift towards male higher variability. So there's actually not much, these are gonad intact humans and mice. There's not much objective evidence that there's actually greater variability. So now the synaptic physiologist no longer has the excuse that they don't want to study females anymore because of the hormones, and it's important. It turns out, a major point is that studying both sexes is required for gender equity. Okay, it's just not okay to study one sex and leave the other sex out. As we're trying to celebrate today, that there is some really interesting stuff going on when you compare the sexes. Now, NIH, another hero is Janine Clayton, who's the director of the NIH Office for Research on Women's Health. And she, together with Francis Collins, wrote this article in Nature in 2014, where they announced that NIH was developing policies that require applicants to report their plans for balancing male and female cells in animals. And then this year, in June, the NIH announced that by next year, if you're writing a grant application, you have to at least talk about why your studies have, why the biological factor of sex is taken into account and why your studies are going to be applicable to both sexes. So we're expecting some change here in the sex bias in biomedical literature. So in the next part of the talk, I want to talk about some just kind of evolutionary ideas about the evolution of XX and XY cells. A fundamental point is that, where do sex differences come from? They come from the sex chromosomes. So in the zygote, and the first day of life, when the zygote forms, the only thing that we know that's different between the male and female are the sex chromosomes. And I state that dogmatically because it might be wrong, but right now, nobody's challenged me, and I've been stating this for several years in talks, bring it on, tell me about another sex difference besides the sex chromosomes. Is there a sex difference in cytoplasm? We don't think there is, but there might be. But right now it's the sex chromosome. So that's where sex differences start, and everything stems from that. Okay. How do sex chromosomes evolve? Well, there are plenty of species in which there are no sex chromosomes, and it's thought that in mammals and birds, for example, a sex determining so in a species that does not have sex chromosomes, on one autosome there arises a sex determining mutation. In the evolution of mammals, that's the mutation of SOX3 into SRY. SRY is the testis determining gene, which is a dominant male determining gene, causes the formation of testes, the development of testes. And so you have the evolution of the X and Y chromosomes from autosomes, and it takes a while. But in birds, it was a different set of genes and that led to the bird sex chromosomes. But once a dominant sex-determining mutation occurs on an autosome, you get the proto-Y chromosome beginning to lose DNA for reasons I'm not going to go into. It's complicated, and I don't even completely understand it myself. Just from what I read in the literature. So the Y chromosome shortens up, loses DNA. The Y chromosome is an old X chromosome. It has a few genes left that are very similar to the X chromosome, but these genes have not recombined and crossed over with the genes in the X chromosome for a long time. So they've separated in their functions, and they are male-specific. They're handed down. A few genes, not very many genes left on the Y chromosome, but the X chromosome's got plenty of genes. It's as big as another chromosome. Now, the loss of genes from the males, the loss of one X chromosome in the male, as the Y genes got shortened, leaves most of the X chromosome in a potentially haploinsufficient state. That is, there's only one copy. And for a lot of genes, that doesn't matter. Okay, it doesn't matter in a lot of genes whether you have one or two copies, but for some, it's really important. Okay, and those genes are dosage sensitive. It matters whether you have one copy or two copies. And for this reason, it's thought that dosage compensation evolved, which is that the female, first of all, there's a selection pressure to increase the expression of genes on the X chromosome in males as they're losing the one copy of the X chromosome, and there's a resulting pressure to decrease the expression of the X chromosome in females. And in mammals, that leads to X inactivation, which is that one X chromosome in the female is inactivated in every somatic cell. So here's the selection pressure I was just talking about. So we have this fact that in both male and female cells, one X chromosome is active. And so it's thought, well, the X chromosome is probably not doing very much to cause sex differences because females express one, males express one. On average, the X chromosome expression is kind of balanced. Now, if it weren't balanced, just think about this for a second, the double dose of X genes in females would cause a maladaptive sex difference in X gene expression. The problem here is that the X genes are interacting with all the autosome genes. The males and females have the same number of autosome genes, but the females, if they had twice as much X stuff as males, the level of conversation between the X genes and the autosomal genes would be off balance in one sex or both sexes. They would never be able to reach an optimum. So if this, we can say, okay, how effective is X inactivation? And here here's a curve measured again from microarray studies, again by the same guy, Ito, where each gene in the microarray study, and this is from mouse, brain, and liver, each gene gets a male to female ratio. So you measure the expression in males, expression in females, calculate a male-female level of expression here, and say, okay, is it higher in males, is it higher in females? You take the log of it and display it on the curve, and you can see that for brain and for liver, there is the modal gene expression is sexual equivalence. The log to the base 2 of 0 is a sex ratio of 1. The log of 1 means that the male genes are twice as high as in females. The log of -1 means that the genes are twice as high in females as in males. And you can see that it's pretty sexually equivalent. There are a few genes that are higher in males, a few genes that are higher in females, but basically it balances out. These are autosomal genes, where everybody's got the same copy. We'll say, well, what about the X chromosome genes? If the X chromosome were not inactivated, then you'd expect the X genes to be out here. That is, to have maybe about twice as high expression in females as males. But the actual data shows that the X chromosome genes are, in fact, as sexually equivalent on average as the autosomal genes. So X inactivation is incredibly effective at pulling that curve of female bias and matching it actually tissue by tissue. Notice that the liver is more sexually dimorphic. There's more sex, there's more male bias and female bias genes in liver than in brain. And this is whole brain, where you're taking out the whole brain and just grinding it up and doing a microarray. So there might be brain regions where there's a lot more sexual dimorphism than is shown here. There's a little bit of more sexual dimorphism on the X chromosome than on the autosomes, but not hugely. So X inactivation is quite effective. But you have to think of X inactivation as a compensatory process. It's a process that makes the female more like the male by counteracting an imbalance that it pre-exists. The imbalance is in the number of X chromosomes. That's a sex bias. Females have 2, males have 1. And the X inactivation comes as another female-specific process that undoes the deleterious effects of this sexual imbalance. So this X inactivation is a part of sexual differentiation, in my view. It's because it only happens in one sex. The female cells spend a lot of time and epigenetic effort to shut off one whole X chromosome in every cell of the body. And males aren't burdened with this at all, okay? So it makes cells different. We don't really understand yet the kind of biasing effects of just having an inactive X chromosome in every cell. But I think that's coming. But there is this evolutionary pressure to make the sexes similar. Now, just as a side point, if you read any article on sexual differentiation of the brain or of any other tissue in the 20th century, nobody, even though X inactivation has been known since the 1960s, nobody ever listed X inactivation as part of sexual differentiation. Why? Because sexual differentiation used to be just reproductive tissues. Do you have a penis or a clitoris? Do you have a uterus or no uterus? Do you have sperm ducts or no sperm ducts? That's what sexual differentiation was about, and all the reviews were about those tissues. But we have to, now we think much more broadly, and I put X inactivation on the list of processes that make cells different by paradoxically making them more similar. But one sex is doing something the other sex is not. And so I actually think of sexual differentiation as kind of this balancing act. Part of sexual differentiation is when males need to do things that females don't, and females need to do things that males don't. Females need to get pregnant, males don't, right? So that makes some structures, and there are things that become adaptive for the female because she has a different niche. She's got to carry the fetus, right? So she has a different niche, and she will be built for that purpose. Males will be built for other purposes, but then there's another part of sexual differentiation, which is just trying to keep the sexes constant when, in fact, they start out not being similar because they have these different X chromosomes. So I don't think that X inactivation has anything to do with did not evolve because of any different niche of males and females. It's just that the female cell is constrained to have 2 X chromosomes and the male cell is constrained to have 1, and that sets up some biases in cellular function from the very beginning. All right, so dosage compensation has profound genome-wide effects on the balance of gene expression in males and females. Just parenthetically, birds and mammals do it differently, and I won't talk about birds anymore. Okay, now the third part of the talk I want to talk about, so who's going to flag me down and shut me up at the right time? It's because I've lost track of time. Okay. The third part of the talk is research that emphasizing stuff that's coming out of our lab in the last 10 to 15 years, because we got onto the idea. I'm a brain sexual differentiationist, as Josh said, for a long time, and in the first at least 20 years of my own thinking about sexual differentiation, it was all about hormones and how do hormones make male and female brains different. But then I got onto the idea, because the hormones in birds, I was studying birds, and hormones in birds weren't explaining the sex differences. So we got onto the idea that the sex chromosomes might also be causing sex difference, and the next part of the talk is the current kind of status of some of our thinking about that. So the framework here, again, starts with the males and females have different diseases. If we can identify, and that means that one sex is protected. So in multiple sclerosis, males are protected. There is no drug that you can give to a multiple sclerosis patient, a female, that's going to alleviate her disease as much as it would if you could just make her male. So the male's got some secret substance, some secret juice, which might be testosterone. But the males have something that medical science has not been able to match in terms of efficacy in blocking or preventing or ameliorating the disease. So if we could only just figure out, I collaborate with a multiple sclerosis specialist, Rhonda Voskuil, she says all we need to do is figure it out, right? Okay, so, and that's what she's been doing and making some incredible progress. And it might be that these sex-specific factors that are protective of disease are actually attractive therapeutic targets because they're not going to kill you. Even giving testosterone to a female patient, which you wouldn't do, isn't going to kill her, okay, because it's a natural product, whereas some drugs that medical profession or pharmaceutical industries might come up with might actually kill you. Testosterone is not likely to do that, even though it's not attractive therapy for other reasons. Changing your sex doesn't usually kill you. So if you could change just one part of being male or part of female, maybe you could. Maybe the hope is to come up with new therapeutic targets. Okay, so at the genetic level, all sex differences result from the X and Y balance. The dogma, but we need a way. If you notice that there's a sex difference in a disease, and you have a mouse model or animal model where you can actually start to manipulate the individual components of being male or individual components of being female, start to manipulate them one by one. You have to have a framework for doing those experiments. The framework from the 20th century is in all the textbooks, and if you ever learned about sexual differentiation in school, you probably learned this. That is, there are 2 processes. The first is sex determination. It occurs first. It's genetic. And that's what determines whether the gonads are male or female. So it begins with the sex chromosomes, but the first thing is a genetic event in mammals, and the second thing is sexual differentiation, which is that the gonads begin secreting hormones which act throughout the body to uniformly masculinize or feminize, depending on the hormone levels. And that's a second process. It's temporally second, and it's hormonal. It's mechanistically different. This is an enormously simple idea, 1-2 genetic hormonal, and it's largely true, but it's not completely true. And the reason is that there are sex differences in the rest of the body before the gonads ever develop. And they're genetic too. They're not hormonal coming from the gonads. So the rest of the body is not uniformly sexually differentiated by hormones. So let's try to find out. And there are sex differences that occur after the gonads develop that aren't downstream of the gonads. All right, so expanding on this model a little bit, it's that SRY The old model is that SRY comes from the Y chromosome. It makes testes develop. The testes secrete testicular hormones, especially testosterone, and that causes masculine development. In the absence of SRY, other genes are turned on, X genes or autosomal genes, to cause ovarian differentiation, which, by the way, blocks testicular differentiation, and the testicular differentiation pathways block ovarian differentiation, so they're kind of reciprocally inhibitory, which is interesting mechanistically. But then female hormones and the lack of male hormones cause feminine body. And now, the reason I don't like this is because there are other pathways, and that's what we're talking about in this third part of the talk. And so a more general formulation, I think, is this, that these XX versus XY differences play out on the gonads, and this is enormously important because it sets up lifelong sex differences in the effects of gonadal hormones, which we can classify as either long-lasting or short-acting and reversible. These are long-lasting and permanent, like the differentiation of a penis is because of the action of testosterone. You take away the testosterone, the penis doesn't go away, so it's a long-term permanent action, whereas a lot of effects of testosterone go away if you take the testosterone away, and those are called activation. So I think Peg might be talking a lot more about this, and I'm going to talk about this, which is that these same X and XY differences play out in non-gonadal tissues because of the unequal expression of X and Y genes, and these are sex chromosome effects. So that's what we're going to talk about. Now you can classify these sex chromosome effects, or these initial, what I call primary sex determining genes. This is actually not the way that people use the word sex determination. These are genes these are mechanisms that make XX and XY cells different, inherent mechanisms. And one is that the XY cells have Y genes that the XX don't, and that can have effects. But the Y chromosome is pretty pathetically small. And the people who have this unfortunate chromosome are worthy of your support. But so SRY is a biggie on that chromosome. It makes testes grow. There are some really important spermatogenesis genes that make males be able to make sperm. And we don't know too much about the other ones as far as their effects on general body phenotypes outside the gonads. Another big one, which is where we're putting our emphasis and money currently, is X dosage. That although X in activation, although X in activation is effective, and there is a big bias in X gene expression before X in activation kicks in early in life. So developmentally that could be important. But in adulthood, there are a bunch of genes in humans and mice that escape X in activation, so are expressed from both X chromosomes. So they cause an inherent bias in expression. So there are some genes that are inherently X genes, not many, that are expressed higher inherently in each XX cell compared to each XY cell. There is a sex difference caused by imprint because only females experience the imprint that comes from the father. And there's something kind of speculative and interesting, exciting, called the heterochromatin sink, and that's the idea that having an inactive X chromosome in each female cell actually sucks away heterochromatizing or factors, epigenetic factors that could be acting on the autosomes, sucks them away in a way that doesn't happen in male cells, and that there's some evidence for that. But okay, so those are 4 different mechanisms. So, but the main problem is, how do you vary the sex chromosomes without changing hormones? Okay, because if you say, I want to study Y genes, well, let's just take a female and give her a Y chromosome and see if it changes her. Well, if you give her a Y chromosome, one of the things that's going to change is that she's going to have testes, and then she's not a female anymore hormonally. So how do we manipulate the sex chromosomes without changing the gonads? And some years ago, I discovered a model that was developed by Paul Burgoyne and Robin Lovell-Badge in London, which we call the 4-core genotypes, which is one of 2 models I'll talk about today. The other one's called XY star, which is a different model, but the first model that we use is the 4-core genotypes. And in this model, you basically take the gonadal sex off of the sex chromosomes so that you can study the sex chromosomes independently of what. And so here's a schematic of the genome. The SRY is on the Y chromosome. It's deleted from the Y chromosome and inserted as a transgene onto, it actually is onto chromosome 3, it turns out. We now know where it is. And so this is a male. It's got all the male genes, but one gene's in the wrong place. And so this animal makes 4 kinds of sperm. It makes X and Y sperm, but the X sperm can either have S or Y or not. The Y sperm can have X or Y or not. So there are 4 kinds of babies. There are XX and XY animals that don't have S or Y and have ovaries, and XX and XY animals that do have S or Y and they have testes. Okay, so it's a 2-by-2 comparison. This is one of our kind of lab logos here. They're either XX or the animal is either XX or XY, and they either have testes or they have ovaries. So we can say, in any trait that's sexually dimorphic, does it matter whether they're XX or XY, or does it matter whether they have male or female gonads and hormones, and or does it actually do both of these factors matter? So, so if we measure the phenotype in all 4 groups, and the phenotype comes out to be the same in the 2 gonadal male groups and different from the 2 gonadal female groups, where the color here is coding for the level of the phenotype, then we conclude that the sex difference is caused by the gonads, and likely it's caused by gonadal secretions or hormones. But if we get the opposite result, which is that the XX animals are both the same and they're different from the XY and they're both the same, then we say it's not gonadal sex because the 2 sexes here are equivalent. They're the average of gold and blue, which are UCLA colors. They're the same here, but these are different, so it has to do with sex chromosomes. Now, I have to do this, which is, if you stare at the slide, this is optical illusion. Peg's seen this before. You stare at the slide for 5 seconds. 5, 4, 3, 2, 1. So how many colors do you see? I'll do it again. So 5, 4, 3, 2, 1. So you're supposed to see 4 colors, except now you can only see 2 because your cerebral cortex got used to it, and there's an after image in your brain, and that is a model of when both factors are important, because then you get 4 colors. That's if you think about it for a minute, which but undergraduates love that optical illusion. So the question is, when we started, would we ever, based on the dogma said you'll never get this because it's all hormones, right? So we were kind of gunning to kind of knock down the dogma, and so we really wanted to find this, right? And the best, I'll mention a few cases now where we think it's really important that the animal is the cells are either XX or XY. One is a collaboration with Karen Rui in the Department of Human Genetics at UCLA, and she's a geneticist and an expert in lipids and metabolism. And this work was done by Suki Chen in my lab and Jenny Link in Karen's lab. So if you just weigh a mouse, the males weigh more than females. This is so we got 4 groups here. They're either XX or XY, and they either have male gonads or female gonads. At weaning, they don't weigh much different, but if you wait a couple, 20 days later, 24 days later, then the males have gotten to be bigger than the females just in body weight. And so because it's the 2 animals with testes that are bigger than the 2 animals with ovaries, we say, okay, that's probably hormones, right? And so the first experiment you want to do is take away the hormones to see if that sex difference goes away. And so here's the graph when you take the hormones away. So the 2 male groups are heavier. On the first day of this graph, week 0, the animals are 75 days old, so they're 2.5 months old. And the males are heavier than the females. If you take out the gonads about a month later, the females have gotten heavier and the males have stopped getting heavier. And that actually, and we think that both male and female hormones made this difference, and the difference goes away. Conclusion that the main sex difference is caused by gonadal secretions, and you could take away the hormones and the effect goes away. So these are what we would classify as reversible activational effects of hormones. And so most of the time, you just stop the experiment there because you just reverse the sex difference. If you're interested in sex differences, you'd go off and study hormones, okay? And is it the testicular hormones? Yeah. Is it the ovarian hormones? Yeah. Both are important. But for bizarre and accidental reasons, we actually kept on the experiment. The reason we did this experiment actually was we were collaborating with somebody in the VA, and the VA has terrible rules of quarantining the animals. We had to quarantine the animals for months before we could study them. And so the animals were this old by the time we actually studied them. And Suki Chen in my lab was studying stress and putting animals in a tube for 30 minutes. It was a mild stress. Their corticosteroid levels go up. We were interested in their cortisol levels and effects on the gastrointestinal tract, stuff like that. But she came back and said, I can't, some of the animals don't fit in the tube. and they're all XX, right? So what happens is the XX animals, after you take out their gonads, get really fat and heavy. And it doesn't matter whether they it actually does secondarily, but both animals that used to have ovaries and used to have testes, they haven't had, this is like 8 months later, they haven't had gonadal hormones for a long time. So they used to have gonadal hormones that make a difference in the XX group, but not in the XY here. But in any case, the big effect here is that there's an XXXY difference. This effect is as big as the hormones were when the hormones were around. So it's not a small effect. And Karen Rui and we have done studies that show that it has really pervasive effects on metabolism. So when the gonads are present, the gonadal hormones dominate. By the way, you say, well, what happened to the sex chromosomes? The animals have always had the sex chromosomes. Do they make a difference? And the answer is, yeah, they did make a difference when the hormones are there. The XX animals are heavier than the XYs in both groups. But it's a smaller effect than the hormone effect. So that's about an 8% body weight difference, whereas the hormone effect's about a 25% body weight difference. So there's a sex chromosome effect riding on top of a hormone effect, so you get 4 colors, not 2. But if you take out the hormones, so you have these kind of balance. And by the way, the female genome makes the animal heavier, but the male hormones makes the animal heavier. So they're acting in opposition to each other, compensating for each other and reducing the effects of each other. They are reducing the effects of each other. So either the hormones are turning off the sex chromosome gene effects, or the sex chromosome genes are turning off the hormone effects, or something. Nobody knows, and it would be interesting to study. But so you have this compensation happening here too. So the sex is okay. I just said all of these things, so I'm going to go on. There is this opposite effect of sex. The female genome makes the animal heavier. The male hormones makes the animal heavier. And this may happen a lot, that you have this compensation and this evolution of the two sexes towards each other by the XX animals stuck with having a second X chromosome. So it might be that the hormones are turning off some of the deleterious effects of having a second X chromosome. You could think about it like that. All right, but the real effect here is on body fat, is that if you do a body composition study, the XX animals have almost twice as much body fat as XY. Genetically, we'd really like to know, are these X genes or are these Y genes doing this effect? And so we have a second model called an XY star model. And they have some weird genetic names, which it would take me half an hour to explain. But it's important to understand that these are very similar to these genotypes. So you have a female with 2 X chromosomes, and you have a male with 2 X chromosomes and a Y chromosome. You have a male with 1 X and 1 Y, and you have a female that's got 1 X chromosome plus an extra pseudoautosomal region. So these animals either have 2 X chromosomes or 1 X chromosome. They either have 1 Y chromosome or 1 or 0. And you can do the same experiment again. At week 0, the animals are 2.5 months old. The males weigh more than the females. You take out their gonads, the sex gonadal effects go away, and the animals with 2 X chromosomes get fat, and the animals with 1 X chromosomes don't. So we don't think it's the Y chromosome. We think it's the X chromosome, and there's genes in the X chromosome. Well, what are the genes? Genes escaping X inactivation are expressed higher in XX and XY cells in a lot of tissues, including these liver and gonadal fat and brain and a whole bunch of other places. And here's some of our big candidates, and we're now studying the dosage effects of these genes on metabolism. What's different about these animals? Well, it turns out that when the animals are active at night, the amount of feeding is about the same. But in several studies, so we've confirmed this several times, when the animals are supposed to be sleeping, the XX animals are eating more than the XY animals. And it's not a big difference in total food consumption. It's just the time of day. In fact, there is no difference in total food consumption. But the time of day at which they eat is different, and that we think might be interesting. So it maps into circadian rhythms of eating. So the animals might get fat very slowly over months and months because they have shifted the time of day that they eat. If you put the animals on a high-fat diet, which if you're a metabolism person is the first thing you think to do, to kind of stress the system metabolically, it just exacerbates the sex chromosome effect. So you get a clean separation of XX and XY difference on a high-fat tissue, high-fat diet, and the tissues of the body respond quite differently to this high-fat diet. So you can just you do a sex chromosome analysis of the animal by looking at its liver after it's been on a high-fat diet. Here's a liver if they're XX, here's a liver if they're XY. These little white things here are fat globules in the liver. So the liver becomes very fatty. It's a precursor in humans for liver cancer. If you measure HDL cholesterol levels in the blood, doesn't matter what kind of diet they're on, doesn't matter whether they have gonads or not. The XX animals have higher HDL cholesterol than the XYs. Okay, so this is a summary of some of the things I've said. It's the number of X chromosomes that has dramatic effects on adiposity and weight gain. 2X is worse than 1. So having a second X chromosome is a problem in this disease model. How much time do I have?
Good. Still have 15, 20 minutes.
All right. I won't take that long. A second case study of the effects of sex chromosomes is a collaboration with Mansoor Eghbali, who's in the Department of Anesthesiology at UCLA. She's an expert on cardiovascular disease and mouse models of cardiovascular disease. So she took our mice to say, okay, she's been studying hormone effects and estrogen effects, which are quite protective. In general, women don't get cardiovascular disease as much as men. And the dominant story in the literature, and there's a lot of work on estrogen effects, is that estrogens are protective. Makes sense, and it's true, okay? But I'm going to show you data that having 2X chromosomes is a problem. So it goes in the wrong direction. And again, it might be a compensatory change here. But, okay, so basically what you do is you take the mouse and give it a heart attack. Now, we take the gonads out before we do this because we don't want the hormones to interfere with we think that unmasks the sex chromosome effects. So these animals are all gonadectomized. You can do this in vivo by ligating the LAD artery, and so you shut off blood supply to the heart for 30 minutes of ischemia, then you release it, and the reperfusion causes injury, which you look at 24 hours later by staining the heart with Evans blue dye. And you can look at the healthy tissue is red, the at-risk tissue is blue, I think, I may have these wrong, but the white tissue is the bad part, okay? So look how much white is here and not so much white here, and these are the 2XX groups. So here's the infarct size measured as a percentage of the area at risk, and the XX animals have greater infarct size than XY. You can also do this in an in vivo model where you take the heart out of the animal and reverse perfuse it with kind of an oxygenated buffer solution, kind of ringers. And here you let the heart stabilize ex vivo for a while, then you give it a 30-minute ischemia, then you let it go for an hour and measure. Here you can measure hemodynamic properties of the heart and the infarct size outside the animal. Here's some traces. You can see these are pressure traces, and the pen, the electronic pen is going up and down here as the heart beats. These are heartbeats. You shut off, here's the period of ischemia, and the heart stops beating. Then you let the perfusate start again, and the heart starts beating again. But the recovery of function after this period of ischemia is worse in XX animals than in XY, replicating the in vivo effect. And again, so I'm not going to show these. It's a bunch of documentation that physiologically the hearts are recovering better if the heart is XY than XX. But the infarct size, again, is worse in XX than XY. So again, we wanted to say, okay, is this an X effect or a Y effect? It's an X effect again. So here's our this other model mouse, which is the XY star model. Animals with 2 X chromosomes. So this is the left ventricle developed pressure. So after reperfusion, if more pressure is better than worse, but the animals with 2 X chromosomes, whether or not they have a Y chromosome, is worse than animals with 1 X chromosome, whether or not they have a Y chromosome. So again, the X chromosome is a problem in this mouse model of disease. Again, the genes that we're interested in and we think might be important are these X escapees. 2 of them are histone demethylases, and one of which Josh Rubin mentioned, which is KDM6A, also called UTX, which is of interest in his lab in characterizing sex differences. So we think that one or more of these genes might be causing this effect. So the third and last case I want to mention of sex chromosome effects is multiple sclerosis. And this is the research of Rhonda Voskuil at UCLA. She's a physician who's the head of the MS Center. And she also does a lot of mouse studies of a model of MS called EAE, or experimental autoimmune encephalomyelitis. And again, you give the mouse the disease by injecting a myelin antigen, which induces an immune reaction to the myelin proteins. And then, you can do it in several different ways. I won't go into the different active and adoptive transfer models. But here is a clinical score kind of result from her lab, where you can rate how poorly the animal is doing when you give it the disease, this EAE disease. So 0 rating means the animal's perfectly healthy. 5 is a dead mouse, and 1 is a slightly impaired mouse. There's just more progressive impairment. And you can see that for about a week after immunization, the animal does just fine, but then the disease, the immune system kicks in, and the animal gets pretty bad, actually, and then recovers. And this actually kind of mimics and mirrors the disease in humans, which is relapsing and remitting. It gets worse on some days and gets better for some weeks. But the XY group, in the end, does better than the XX group. These happen to be males. You do the same thing in females in this 4 core genotypes model. You get the same result. If you look at neuropathology, the XX females are worse than XY again. So in general, and using a different model, which is a model of SLE or lupus, which is an extremely sexually dimorphic disease in humans. This is a model in which pristane, which is an adjuvant, is injected in the mouse, and they eventually die from this. And here's some death curves of SLE modeled mortality, and the XX animals, again, these are both gonadal males, are doing better than the XY in terms of how fast they die. All right, so we've gone through these 4 points. I've shown you in the last part of the talk, which was emphasizing number 3 here, that XX and the cell is different in its response to disease. The animal's different, tissues are different depending on whether they have 1 X chromosome or 2. So we're eager to find the genes that are causing this so that we can begin to study, among other things, how those genes interact with the hormones, which are also influencing the same diseases. So I'm not saying that these diseases are all sex chromosome, not by any stretch of the imagination at all. Each of these diseases has potent effects of gonadal hormones. So we've tried to remove those to reveal the X and Y effects. And this is our general model, which I showed before. I won't go into it again. And so I want to thank the people who did the research in my lab. Over the last decade or more, Yuichiro Ito and Suki Chen have been the kind of pillars of my lab. And these other people, these are all the people who've taken care of the mice, which is a big deal in our lab. And Shane and Miriam have done projects that I haven't actually talked about today. We have a wonderful set of collaborators at UCLA, including Karen Rui, Mansour Iqbali, Eric Verlaine, Rhonda Voskuil, and we get almost all of our mice originally from Paul Burgoyne, and so he's been my main guru and advisor for almost 20 years now. We have a group of colleagues that are highly interactive, and we have a T32 NIH grant, which is supported our students for the last 30 years. And so this is a great group of educational faculty teaching students and postdocs about sex differences. And we really appreciate the support from the NIH, which has enabled all this work, and from other places as well in various pilot grants. So thanks very much.